All right. So, as you saw in the mealworm investigation, animals eat. Our mealworms ate potatoes, cows eat grass, that baby you see there is eating a cucumber, and Christian Bale eats tacos. But what happens once that food enters an animal's body? Today we're going to investigate what happens to food at the atomic molecular scale. In this activity, we're going to be investigating what happens when a cow eats. Cows, like all other animals, have inputs and outputs. Food and oxygen go in, and feces, or poop. Urine and carbon dioxide come out. We are going to look at what happens inside the cow and how inputs turn into outputs. First, let's get the poop part out of the way. Not everything we eat is digested. Our digestive systems can't break down cellulose, and that leaves our bodies as feces or poop. 50% of poop is undigested food, and the other 50% is bacteria. So here you can see that cellulose getting converted into waste, into poop. It goes again. So other than poop, some of the food a cow eats sticks around. You can see here that the majority of what a cow eats is water and carbohydrates. These go into a cow and become part of the cow. The cow is made mostly of water, protein, and fat. Then some materials leave the cow. The cow breathes out water and carbon dioxide and poops out water and carbohydrates. We're going to be looking most closely at this part here, how the food gets turned into protein and fat. Here are the four main questions we're going to try and answer in this activity. 1, what is food made of? 2, what are animals made of? 3, how can cows get food to a cell in its leg? And 4, how can cows use this food to build their bodies? So I'm going to pause here for you to answer some questions. You can go back and rewatch any part of this video you need in order to answer the questions. All right, so let's review. Food is mostly water and large organic molecules. Until now, we've been using shapes to represent the different food molecules for fats, carbohydrates, and lipids. For example, we used hexagons to represent carbohydrates and the glucoses in carbohydrates. We used rectangles to represent the glycogen and glycerol and fatty acids in fats, and we used a variety of shapes to represent the monomers in proteins. But starting now, we're going to look at these molecules at the atomic molecular scale, so you can keep track of what atoms are in the food molecules and where the high energy bonds are. So here you can see all of the high energy bonds are highlighted in yellow, anything between a carbon and a carbon, or a carbon and a hydrogen. So since a cow is mostly fat and protein, how does a cow make fats and proteins from the food they eat? So that's the big question we're trying to answer here. We are going to trace the organic food molecules through a cow. Food starts outside the cow, at its mouth, so put a penny or another marker there on your poster to mark where these food molecules begin. As you can see, we will be following the food through several different places, including the stomach, intestines, heart, leg muscle, and lungs. The first step after food is eaten is digestion. This takes place in the stomach and the small intestine of the cow. We will deal with biosynthesis and cellular respiration in future activities. So move your marker to the stomach and small intestines part of the cow. This is where we're going to spend our first chunk of time. During digestion, large organic molecules are broken down into small organic molecules. For example, starch is going to be broken down into individual glucose molecules here. I'm going to pause here for a couple more questions for you to answer. As we saw at the beginning of this lesson, some molecules cannot be broken down, like cellulose. These molecules leave our bodies as feces. We'll be focusing on the other food molecules from here on out: proteins, starches, and fats. I just want to remind you of the rules about how atoms bond together to form molecules. This will be useful as we move into digestion and later into biosynthesis. First, each atom has a preferred number of bonds it likes to make. Carbon likes to make 4 bonds, oxygen makes 2, and hydrogen makes 1 bond. Oxygen atoms do not bond to other oxygen atoms if they can bond to carbon or oxygen instead. And remember those high energy bonds. The bonds between two carbons or a carbon and a hydrogen, those have high chemical energy. Other bonds have low chemical energy. Wrong way. Other bonds have low chemical energy, like carbon-oxygen and oxygen-hydrogen bonds. So we're going to be focusing on those high energy ones instead, the carbon-carbon and carbon-hydrogen bonds, and represent them with a bright yellow line between them. So now you're actually going to start digesting molecules. Find your card with proteins on it. It's the one with red nitrogen atoms. Take your scissors and cut along the dotted lines. Notice that when you do this, you leave a carbon atom with only 3 bonds and a nitrogen atom with only 2 bonds. Neither of these atoms is happy. Carbon wants 4 bonds and nitrogen wants 3. In order to fill those empty bonds, you will need to cut water molecules apart. Cut each of your 3 water molecules along the dotted lines. Attach the OH to the carbon atom and the H to the nitrogen atom. I'm going to pause here so you can do your cutting and taping and answer a couple of questions. Just to back up a little bit, back to this one here. We used water. We broke apart water to make this happen. So we started off with 3 water molecules. When those went in, the water molecules were used to take up those extra bonds. When you use water to break apart a molecule, that's called hydrolysis. Hydro means water and lysis means to break. So when you're using water to break something apart, that's called hydrolysis. All right, now you are going to do the same thing for starch. Find your card with a starch molecule. This is the one with hexagons. Cut along the dotted lines to break your starch into 3 glucose molecules. Again, you are left with a carbon atom with only 3 bonds and an oxygen atom with only 1 bond. You'll need to cut apart water to satisfy those bonds. Attach the OH to the carbon in the glucose molecule and the H to the oxygen that was left in the glucose molecule. I'm going to pause here to allow you to cut and tape and answer a couple of questions. All right, here we're going to watch again those carbon atoms. We're going to watch our digested starch and see what happens when it goes through hydrolysis. So we have our starch and our water, goes through a chemical change. Wiggle, wiggle, and out comes 4 individual glucose molecules. And we use those water molecules, we broke them apart to satisfy all of those bonds. Here we're going to keep track of the chemical energy and the carbon atoms. And if you were to actually count them up, you would see that you end up with the exact same number of carbon atoms and the exact same number of high energy bonds as you started with. So the carbon atoms stay in organic molecules with high energy bonds. Finally, you're going to digest the fat molecule using hydrolysis. Cut along the dotted lines to separate the glycerol from the 3 fatty acids. Then use your water molecules to make sure that the carbons end up with 4 bonds each and the oxygen atoms end up with 2. Remember that oxygen doesn't bond to another oxygen if it can bond to hydrogen instead. I'm going to pause here so you can cut, tape, and answer the questions before we move on. All right, here's the animation of hydrolysis of fat. Like before, we can see that the number of carbon atoms stays the same and chemical energy is conserved throughout this change. So here's our fat getting broken down through hydrolysis, breaking apart those water molecules, and we end up with 3 fatty acids and 1 glycerol. And following the carbon and the energy, excuse me. Again, we see that the carbon atoms are conserved and the chemical energy is conserved. So those carbon atoms are still in high energy, organic molecules with high energy bonds. So now you have a bunch of digested monomers, glucose, glycerols, fatty acids, and amino acids. At this point, all the chemical energy we started with is still chemical energy. No high energy bonds were broken. So where do these digested monomers go? What happens next? We're going to pause here to answer a few more questions. Once the polymers are broken apart into their individual monomers, the pieces are small enough to be transported through the bloodstream to different parts of the animal. Here you can see that blood carries the monomers to the leg muscle of the cow. In the next lesson, we will investigate what happens to these molecules once they arrive in the cell, a process called biosynthesis. Finally, to relate this to the cartoons of the molecules you have used in previous lessons, you can see that cell growth starts with digestion of the molecules, or breaking the polymers into monomers. Then, these monomers are transported through the cell membrane into animal cells. Tomorrow, we'll see what happens once they get there. So this is the end of the digestion part of what happens to your food once you eat it. There's going to be 4 more questions for you to answer, and then tomorrow we're going to find out what happens inside that cell.